Well, welcome to the Armidale Genetics Summer Course for 2026. We're very excited to have all of you here in Armidale. We've got 42 people visiting the campus and then about another 20 people joining here from UNE. So for those that are visiting, welcome to Armidale. We're really excited to have you here for this week. So we're doing things a little bit differently this year. So this year, the summer course is supported by the ARC Training Centre for Predictive Breeding. And what that means is for a lot of the people attending here would already know that and are a part of the centre, but for those that aren't, it's a training centre that provides training for postdocs and PhD students in quantitative genetics with a view to creating the next generation of breeders. And so that ties in really, really nicely to what we're going to be doing this week with Agustín. And so we thought it was a great opportunity to kick off the summer course, kind of a bit like an opening ceremony, doing a seminar for the centre. And so that's what we're going to be doing for the next hour and a half. So I'll begin by welcoming Agustín Blasco. So Agustín's travelled from the University of Valencia in Spain, and I'll talk a little bit about that in a second. My name's Dominic Waters, so I'm a lecturer here at the University of New England, and with Julius van der Werf and Tassan, we'll be helping to coordinate the summer course for this year. So if you have any questions, you can talk to one of us. We'll give you some more information about the actual summer course, but for now we'll just proceed with the seminar for the training centre. So I'll start with an acknowledgement of country. So the ARC Training Centre in Predictive Breeding acknowledges the traditional owners and their custodianship on the lands on which we meet. And so here in Armidale, that's the Anawan people. We pay our respects to their ancestors and descendants who continue cultural and spiritual connections to country, and we recognise their valuable contributions to Australian and global society. So just for some housekeeping, the seminar is scheduled from 9 o'clock to 10:30 with a question and answer session probably from about 10 past 10. So hold your questions to the end. And if you're online, please use the Q&A tab to submit your questions. Don't use the chat function. So today's speaker is also the deliverer of the course this week, Professor Agustín Blasco from the Institute of Animal Science and Technology, the University of Valencia. So Agustín's had a long career in quantitative genetics focused on animal breeding, particularly in pigs and rabbits and also in beef cattle. And so Agustín's done lots of interesting work, particularly in rabbits with selection experiments, looking at traits like resilience and feed efficiency. And so for basically this week, Agustín will be imparting his wisdom to us. But what this is an opportunity to talk about the work that Agustín's done throughout his career. So with that, I'll invite Agustín up for the seminar.
Okay. Thanks very much. Thank you very much for the organizer for inviting me to be in Australia once more. I have been here, I think, twice or 3 times before, but I always like to come here. And let me check whether that works. Yeah, that works. And well, I was asked, to talk about what. I have receiving emails from 3 different people asking me, oh, you can talk about your career, you can talk about rabbits, you can talk about the research in Valencia. I said, well, in that case, it's the best is to talk what you want to talk, and probably the success will be more guaranteed. I will talk about everything, mainly about what we do in the Valencia University. There are 2 universities in Valencia. We are the Polytechnic University, which is a university dedicated to technical engineering in general, and for example, civil engineering, because in Spain, agricultural engineering is about a part of agricultural science, and a part about civil engineering for agriculture. We have to learn both. The degree is about 5 years. Now it's been changing because of the Bologna, rearrangement of studies in Europe. But when I studied, it was 5 years or 6 years degree, a long degree in a difficult career in which not many people could, obtain finally the title. And at that time, the engineering was considered a kind. I mean, the university when I started was really a real disaster. It was like a kind of academy in which people were talking there, things that they have read in books and things like that. Nowadays, the agricultural school of my university, of the Valencia University, is among the first 100 agricultural schools in the world, according to the Shanghai ranking. But when I started my career, I had just a piece of paper, a pen, and a table, and people say, you should do research. We received only the Journal of Animal Science and the CABI abstracts to ask for, papers and so. So I come from the deep hole, and I'm very proud of that. We have now farms, labs, and we are highly considered in our world. And about Valencia, well, Valencia was chosen by an international committee of students, by doing some kind of inquiries. The best place for foreigner students to be in, because it's a town with a very nice climate, with the exception of July and August, that we have 35 degrees or so of temperature, but the rest of the year, during winter, we have 12, 15 degrees, and it's always sunny. I mean, the weather is boring, good, sunny, good climate, always the same. And the town is a 15th century, well, it's a town that in the 15th century was the capital of the Mediterranean, which means that it's a town plenty of Gothic buildings, civil Gothic buildings and religious ones, both. Because at that time, Valencia was much more important than Barcelona, and it was really the town that was governing the Mediterranean area. The Valencian kingdom had even colonies in Greece. Athens was one of the colonies, and they went there, found a strange church that they consecrated, and it was the Parthenon. So, I mean, finally, Valencia was an important zone at that time. Apart from that, Valencia is so important because it's been promoting modern architecture. The Valencian architect, Santiago Calatrava, which is very well known in the world, he has made, for example, the metro in the zero zone in New York, and things like that, he has created an opera house, which is spectacular, not as much as the Sydney one. I went, on Friday to an opera in the Sydney Opera House, and it's really probably the most beautiful opera house in the world. But there is modern architecture as well, and we have things that doesn't depend on us. Probably because of that, Valencia is so nice. The climate doesn't depend on us. If the climate would depend on us, probably the Norwegians and Swedish will have the sun and we have the clouds and cold weather and so, but it doesn't depend on us. So we have a nice climate, and we have a wonderful beach that doesn't depend on us. It was there when we arrived, so it's great. We have things that, makes Valencia a nice place to stay in. I start, when normally people finish with the crew, with the people that are working with them, I start now saying which people accompany me along my career, are accompanying me, because they are the key of my success and the key of the success that we have, In this crew, we have 3 professors, 3 senior lecturers, 4 postdocs, and 4 PhD students. So it's interesting because to have a good team is one of the secrecies of the success. When you have a good team and each one is working with related topics, and can stimulate each other, that works very well. And for some reason that I never understood, the staff, which means the professors and lecturers working with me, have been always women. I didn't choose that. I mean, it's just a matter of chance. I finally was complaining because I didn't know how to address properly to a man. I was not have the habit, But I mean, I had the great luck that all of them are excellent people. And in the case of Noelia Ibanez, she's the heritator. So she's a professor that is heritating because I am retired now. In September, I was 70. although I still look like 69, but in Spain, the law obliges you to be retired. So now it's Noelia who is leading, the group. Well, which research line do we have? Well, in front of our building, because I didn't say you that this is our building. Our buildings are not like here, extended, but are, we do not have as much land as you have. We have to have several flats, to put the labs and so. And the farms are in the other side of the street. Of course, it is prohibited to have farms in the urban areas, but for lighting problems, we have lighting solutions. There are not farms, they are laboratories of quantitative genetics, and they allow us, to have we produce about 50,000 rabbits per year in these farms, but they are still laboratories of genetic quantitative genetics, activity and things like that, So, and we have been working with rabbits, with pigs and with cattle. Pigs and cattle through collaborations. We do not have farms of pigs and cattle. We have several farms. We have about 1,000 females of rabbits, in farms that are our property. And we have them near us. But we don't have pigs and cattle. Along my career, I've been collaborating with people, for example, at what is now called Roslin Institutes or in Jouy-en-Josas, with people I've been working with people in selection for ovulation rate in pigs, or selection for parental survival in pigs, or selection for uterine capacity in pigs. That was really an enormous, they had an enormous amount of work to do that, in Edinburgh. But we didn't have pigs. And now in pigs, we are working in genetics of growth and efficiency through companies that are interested in that, we work in that, and they gave us facilities for doing this. They are using facilities, and we're analyzing the data that the facilities produces, etc. And we are handling the selection of the Iberian pig. The Iberian pig is a local breed of people. There are several local breeds of pigs called Iberian pig. I'm not talking about breeds, because breeds is an irrelevant concept. A breed has not any biological meaning or any technical meaning. But as the Australian head in FAO, when I was working at FAO, said once, a breed is a breed if there is enough people saying that this is a breed. So I mean, but we have some breeds between variables about, and we are handling the selection program. We have to produce higher litter size. They have very little, about 6-7 liter size. They have a lot of fat. We cannot reduce too much the fat because it's a factor of quality. But we're dealing, with the programs in Spain. They are far away from Valencia, and they are in the poor areas of Valencia in the west. But and with cattle, we are collaborating as well, with people in Edinburgh, in the School of Agriculture, in experiments that are made with dairy cattle and beef cattle, because we are now deeply in the microbiomics world, and I will tell you a bit, about our conclusions about this. But our cattle work is also through collaborations. Each one of my colleagues is in charge of that. So I will talk about our research in rabbits. What have we done with rabbits? Well, we developed commercial lines, because we are not a faculty of biology, we are a faculty of agriculture. So it's intended that our work has some relationship with what society needs, So, and this is why we were developing commercial lines. And we have been working in the genetics of litter site components and resilience and longevity, and the growth and feed efficiency and carcass and meat quality. We are using rabbits not only as a model, as polite, but as a commercial species, and trying to investigate all the things related to that. But why rabbits? That's a good question. Why are we using rabbits? Bill Hill was asking me one day, saying, well, listen, I would say, why you can use mouse or you can use rats, why are you using rabbits? Well, there are several reasons. The first reason by using rabbits is that we eat them, I mean, we eat about 50 million rabbits per year. I went to the house of Julius, last night, and I looked, that he had the starters. I mean, the starters walking. But apparently, they do not consider the rabbit as a kind of a starter, for a meal. They consider they say you have the starter walking on the floor, but no, well, in Spain we eat rabbits. I mean, it's a most common thing. It's not very popular. I mean, I suppose like kangaroo here or things like that, but it's about 1 kilo of rabbit per person per year, considering babies as well, just born babies, and so. So it's in fact, it's more than 1 kilo. It's about 2 kilos per year for a normally developed person. So and there is a Valencian dish called paella, which is made with chicken and rabbit. So if you go to Valencia sometimes and ask for a paella, and you say, oh, such a peculiar type of chicken I am eating, no, it's rabbit. I mean, but apart from that, rabbit is a good animal model as well. So we can generalize our work to other species. Why? Well, first because it's cheap. You say, oh well, this is not a scientific reason. It is. People working in science know how important it is that things are cheap. The second is that it's easy to collect the data. Collecting data in dairy cattle or collecting data in pigs or things like that is a problem. You have to have, devices, to when you are operating a female and with a rabbit. Rabbit is not as small as a rat that you have to have very careful, and it's difficult to operate them. And it's not as big as a sow or things like that. So has the right size, for working that comfortable with it. The reproductive data are similar to those of pigs, a bit lower than that, 12 liter size, in a crossed rabbit. And rabbit has useful physiological peculiarities. For example, the short generation interval. It is 6 months generation interval. With 6 months generation interval, my friends of beef cattle, dream about that. Well, we have a 6-year generation interval. So for us, it's easy, in few years' time, to have selection lines, that we can examine later. And ovulation is induced by mating. That would be horrible in humans. But in rabbits, it has some advantages, because you know exactly when fecundation takes place. And you can do very accurate, studies about embryo development. The other thing is that ovulation and embryos survival can be recorded by laparoscopy. We developed a laparoscopy technique in which we could observe not only the corporal uterus, but we can observe as well the embryos. I will show you the uterus with embryos without damaging litter size, doing that at day 12 of gestation, at the middle of gestation. So the idea that we can have, in pigs you can't. In pigs you have to put your hand to count, how many fetuses you have there. We can just observe by laparoscopy how many fetuses we have without damaging litter size. So we have the implanted embryos, ovulation rate, and litter size, and the embryo mortality and fetus mortality in the same animal. The other thing is that there is no embryo migration between uterine horns. And that's very interesting, because in that case, I will show you that we can use 2 uterine horns separately for embryo transfers, and we can do wonderful things with that. The embryo cryopreservation is easy. I mean, it's not difficult. And rabbits have secotrophy. Rabbits take the feces directly from the anus and recycle that. Now, we have 2 types of feces: the feces that are going to be recycled and the feces that are yet recycled. The feces that are going to be recycled are protected by a mucus protein. So we have really an ambassador of what happens in the intestine without the need of opening the animal, which is great, because you have just to collect these feces, just avoiding that they eat like directly, and you can have, several moments in the rabbit, in the same rabbit, you can have microbiomic studies and so, and this is an advantage, of the rabbit. And of course, unless you are from Senegal, when I was working at FAO, we were trying to implement rabbits in Senegal, but they didn't want. They wanted to eat the Senegal rat, which is a big rat, that has unfortunately a very small litter size. And we had to build up, farms for Senegal rats. But unless you are from Senegal, people normally do not eat rats. So we can do meat quality studies with rabbits and carcass quality studies with rabbits. And of course, at the end of the day, it makes money. I am not maintaining. You think that to maintain a lab mouse is easy. It's very expensive, but to maintain a lab, farms, a lab, for rabbits is easy because they pay themselves. And they pay sometimes our travels to congresses and things like that. When people say, oh, you are not presenting anything, no, I'm being paid by the rabbits. Several times I travel and so, or I buy a computer paid by the rabbits and things like that. So it's quite useful. So let's start. We will start with the development of commercial lines. Which lines have we developed? Well, how is organized the rabbit breeding? Rabbit breeding is organized like pig breeding, but in a more simplified way. We have 2 lines selected by litter size to produce the cross of a male that popularly is called like hybrids. There is no hybrids because there are no hybrids in animals, and there are no hybrids in animals because there are no pure lines in animals. This is the reason. Not having pure lines in animals, you cannot have hybrid in animals. So, and a line selected for growth, which is the terminal sire. So it's quite similar with pigs, but without measuring back fat or whatever. Now we are developing a line selected for feed efficiency, in which we are recording consumption as well. But by the moment, this is what we have in general. There are some in the world. There are some small modifications to that, but this is the top. And the waste nucleus, multipliers, and commercial farms. Okay? Just as in pigs. We have 4 nucleuses in Spain that are collaborating with us. We send to them our lines, the lines that we developed, and we are now handling the program. At the beginning, it was more complicated. It was by fax. For the young people, it was a primitive instrument in which you put, some data in one side and you receive a piece of paper in the other side, with the data and with that, But now, of course, we are using the internet and computers and so, but we have 4 nucleus and we are dealing with their program, and our technicians, visit them every 6 months, or a bit less, probably every 5 months, to see that everything works well, And these nucleuses have their own multipliers, so we are the responsible of a large part of the rabbit genetics in Spain. They have their web page, in which they can, you can, if you are a farmer, order from one of the nucleuses, the things, and that works like in a company. So there are companies, private companies. Okay? So which lines do we have? We have synthetic lines originally made by crossing commercial hybrids. Can you cross commercial hybrids? Of course, because there are no hybrids in animals. So because of that, we can cross them, and with them to construct new pure lines. You have 3 generations of random mating, to get linkage equilibrium and to avoid the Volmer effect, things like that, and you have the new lines. The new lines are called A, V, R, and E, and so because of the color of the card that we are using. Because at the beginning, the rabbits, as we were crossed for many lines, were now the color white has been fixed, but at the beginning we had rabbits for many colors. And the thing is that farmers complained, oh, this is from another breed. No, this is not from another breed because the color of the card is the same. And the A means amarillo in Spanish, yellow. V means verde in Spanish, green. So the lines are named by colors of the card that we are using to identify them. In order to rid out of the mind of the farmers the idea of the breed, of the morphology. And so at the beginning, they wanted us, as well to have a genealogical book. We said it's not needed for rabbits, I mean, it's not needed in general for any species, but it's not needed for rabbits. We do not need this type of things. Morphology is not considered at all, and we are talking only about production, okay? So we have an R line selected for growth rate. The new line, E from efficiency, selected for efficiency. And we have a divergent experiment selecting for resilience. Not really for resilience, we are selecting for variability of environmental litter size, But one day we discovered, we were selecting that, we are in the 14th generation of selection or something like that, But once we discovered that the animals that have, for example, so I will tell you that later, but some animals produce a constant amount, more or less, of rabbits in the parities, and some vary very much. In one parity has 8 kids, in the other 2 kids, in the other 15 kids, and the others, and there are other females that are civil servants, 10. So why we presumed that the animals, with more homogeneous litter size, will be more resistant to illnesses and to stress. And the reason, because animals have a lot of different litter sizes, because they were more sensitive to stress and to illnesses and so on. It was what happened, in fact. We had 2 different lines. And the homogeneous line is the line that is being commercialized because it's more resilient. But it was great because we didn't know that we were working for resilience. Resilience is a new word. We were working before for reducing, for homogeneous parity, but we are happy to know that we are very modern and we are now working for resilience. So, oh, sorry. Yeah, I was saying that. Well, and there are 2 lines more. The hyper-prolific line that was founded, as we have so many farms, multipliers and commercial farms, we know what happens in Spain with rabbits. So in some place, there is a female that has an enormous litter size. We recover embryos from that female. And we constructed a line that is called hyper-prolific. It's not hyper-prolific, it's a bit more prolific than the other ones, But we constructed that, and we did that with longevity as well. There are a farm in the north of Spain in which a female has had 30 parities. Normally, females have 5 parities as an average. If you have a female with 30 parities, this female has passed everything through her life. We recover embryos from this female and bring that to Valencia. And we recover embryos for all of these females and created a line. I wanted to call that the Rambo line, but they didn't allow me to do that because it was the line of strong females that, passed through everything, and could survive to any circumstance, But finally, we constructed this line that has a special longevity, which is important as well for resilience. So, about 25 million of rabbits, about half of the production in Spain, come from our lines. And we have exported our lines as well to Portugal, Egypt, Turkey, Saudi Arabia, and Uruguay. I was visiting as well China and Cuba, and talking with the Minister of Agriculture in Cuba and in China as well. And in these 2 countries, by bureaucratical reasons, we couldn't implement our nucleus of selection. And it's really poor because they have the knowledge, they have the potential, they could. But the problem is that people knowing how to do the embryo transfers were in a different region from the people having the nucleus of selection. And, 2 different regions in China is like North Korea and South Korea. They are neighbors, but they do not have a relationship between them. In Cuba, the person who was in charge of the nucleus of selections, for rabbits, didn't want the university people telling him which animals have been mated and how. So because they were from another ministry, that it worse even than being North and South Korea. They are more isolated. So it was impossible to do that. It's quite a pity. But we have exported that to this, and we have nucleuses there that work well. Well, let's go to the research lines. These are some of our farms that we have just beside our building at the university, in which we keep the amount, a bit more than 1,000 females, 1,200 females, more or less. As I told you, we produce about 50,000 rabbits per year. We have been working in genetics of growth and feed efficiency, litter size, meat quality, resilience, and longevity, and the development of commercial lines. So now, this is just to start the new point, but this journal, World Rabbit Science, is a Q2, Q3 journal. Normally, it's been in the Q2 for many, many years. And it's important because, can you think, this is a journal that we edit at the university. We publish that in our department, in our university. Can you believe that a journal dedicated to rabbits can be between the 50 best journals, more quoted journals in the world? That happened for many years. And now we're in the Q3, but I found that surprising, that a journal talking about rabbits, can be so highly quoted. The only answer is because the quality of the works was quite good. Well, let us start with genetics of litter size and its components. We have two lines, E and V, selected for litter size at winning. At winning, mainly because, the problem is not to have many, high litters. The problem is to have high litters, that arrive winning. And we have more than 50 and 40 generations, respectively, of selection of that. It's a long-term experiment. And we have genetics of the components of litter size. I will tell you why later, but we had a line selected for ovulation rate and two divergent lines selected for uterine capacity in the years in which uterine capacity were popular. So this is the results of the genetics of one of the results, of litter size. When I was in '86 in Edinburgh working with Robin Thompson produced a paper that, worried us quite a bit, because he said, well, listen, the genetic trends you get depend on the genetic parameters you put in. So if you do not have any heritability and genetic determination of the trait, and phenotypically you observe nothing, but you put a heritability of 0.20, you will get a high genetic trend and a low environmental trend. So, and at that time, when genetic trends were starting to be discovered, that was a great excuse. Every geneticist were telling the farmers, listen, you do not observe the improvement, but this is because I'm here, because your environment is going worse and worse. Just because I'm here, the genetics is getting better and compensates. Well, what happened is that we didn't have any young observation. We were using complex models, including dominance, including inbreeding, considering some environmental effects as random and not as fixed. And by using complex models, we finally got this picture. We had a small genetic trend. The environmental trend, you cannot see it because it's confounded with the black zero. Because we obtained that the environment was more or less the same as we expected, and the genetic trend was going better, but very slowly. Very slowly means that we have one rabbit every 10 generations or 12 generations, which is not a great success, I must admit. So what can you do? We said, well, let's examine litter size components to know whether we can have a better job or not. So have a look to what an uterus of rabbit is. This is one uterine horn, plenty of fetuses, and the other one is empty. And this is one of our colleagues performing a laparoscopy in the screen. And so it's a very easy operation, and a rapid one, not damaging the female. And as you can see, by laparoscopy, you can count the number of fetuses. And well, we use that, for one line selecting for ovulation rate and a divergent experiment for uterine capacity. So if we know that ovulation is made by mating, the implantation is around day number 7, so the laparoscopy is at day number 12, more or less, when we record both traits, and then we record the traits as birth. We have this information. This is the results of our ovulation rate It's one of the things, discussing once with my friend John Hickey, that he was saying, oh, well, we can perform in embryos a lot of genetic edition. We can change about 60 SNPs there. And I said, John, you think that you go to an embryo, you move one of the SNPs, one of the genes, and the rest is performing exactly the same, and is waiting you to finish, what we are doing with that, It's not going, this is not going to happen, I mean, one of the things that I learned when I was working in Edinburgh with lambs, when I was working in Paris with pigs, is that the organism has a big word governing them, homeostasis. That means that if you pull something in the organism, normally the organism reacts to compensate it. So we were augmenting embryo mortality as much as we were selecting for ovulation rate. So finally, litter size at birth or at weaning was more or less the same, although we incremented ovulation rate. And although, because there is no embryo transmigration, you can think, well, listen, if one uterine horn had 7 ova and the other 4, if I increment ovulation rates, I can feel the other uterine horn. No mind which arguments we used. When we pulled ovulation, the organism reacted, incrementing embryo mortality. And about uterine capacity, capacity is not capacity in the sense of volume capacities. Capacity is the sense of you're capable, you're able to bring fetuses to the end of gestation. If you remove one of the ovaries, by this homeostatic reaction, the other ovary reacts and duplicates the ovulation rate. This kind of wonderful homeostasis in which you do a thing and the organism reacts, By duplicating ovulation rate, what we have is a uterine horn which is completely full of embryos, and the other one is empty because there is no fetal transmigration between uterine horns. So we have a full uterine horn and an empty uterine horn. So the idea was that the uterine horn would be as full as possible. So the females having the highest uterine capacity would have also the highest litter size with the 2 uterine horns working. And we checked that as well, of course. The idea was to have 10 generations of selection for and against that. The great thing about divergent selection experiments is that later you have different populations that only differ by genes, by genetics, because they live together, so you can compare both, and says that the only thing. So this is interesting because once I was in a meeting, I received a national prize of genetics in Spain in a competition, if you want, in a meeting with their biologists, agricultural people, doctors, etc. And I said, well, you doctors have much more money than we have much more facilities, but your experimental material is horrible because you work with humans and you cannot slaughter them, you cannot organize the matings, you cannot, whereas you, with my, experiments or divergent selection, I can do marvelous things that you are only dreaming about that. The nice things in rabbits is that you can freeze embryos and thaw them later. So you can have control populations, because a control population after 10 generations of selection is a population that has been selected, you do not know, in which direction, because genetic drift acts. But it has been, frozen and thawed, the genetic drift is minimal. So we compare these figures, 78 animals, 64 and 69 animals, for the 3 populations to know which was the answer to that. And we compare that with the genetic trends that we have. And if what we obtained at the end of the experiment was the same as with the genetic trends, we were validating the model in some ways. This is what the genetic trend said. The experiment was not really successful, but it was successful because we had more, the difference between both lines were quite high. The problem, what we observed is that the main differences were obtained in the first generation of selection. That can happen by several reasons, and one of them is because we have some major gene there, so which invited us to work a bit more on that. Okay? So we started in trying to know much better which were the quality of the embryos, and because we can recover embryos easily by laparoscopy as well in rabbits, and we started knowing, well, let's do some expression analysis and so, but the problem with expression analysis, we have a problem, people working with computers, MC and things like that, is that we do not have proper random generation numbers, because they are not random generation numbers in computers, really. They are artificially random, they are uncorrelated. I suggested once that the best is to take the results of most papers on expression, because they emulate very well random generation, generators, So people do this type of expression, papers saying, what have you found? Oh, I found the things that I expected to find, the usual suspicious people here, and things that I do not know why they are there. There are. What we did is to say, well, when the differences between lines takes place, and we observed that at day 1 of gestation, we were slaughtering females at that time, So females are cheap, we can slaughter a lot of them. So the difference were about 0.4 embryos. At day number 2, the difference still was 0.8 embryos, but at day number 3, the difference was 1 embryo, and day 2.5, 1 embryo. So we decided, well, great, we will do the analysis in day 2.5, which means that, of course, it can happen is that the genes were activated before, and we are observing the end of the scale of activation. But we did that. We were trying to be as much as accurate as possible for our work. And we discovered that uteroglobin, that was a kind of candidate genes or progesterone or oviductin or insulin-like growth factor, could be possible candidate genes for that. But finally, we discovered 2 of these genes that were having an important role in that. Why didn't follow, with this? Because at that time, molecular genetics was expensive, not like now that everything is cheaper and cheaper. And we were deciding, to offer that, to other colleagues and continue our research in other ways, But we didn't want to invest lots of money in that, But they were interesting results, I think. One thing that we did, and we have done with several divergent selection experiments, not only with this one, is to take the high line and the low line. These are the uterine horns of the high line. And these are the uterine horns of the low line. So you can implement embryos of the low line in one uterine horn and embryos of the high line in the other uterine horn. And you can do that in the high line and in the low line. So finally, what you get is the importance of the embryo, the importance of the female, and the interactions. So the problem is that if this is accompanied by any spectacular results, it would be great, but our results were that the embryos had an effect, females had an effect, and interactions as well, which means that the, high embryos in the high line had an interaction, a positive interaction, so nature is complex, but at least we could determine it a bit, the responsibility of each one. And we have done that, in several lines, taking advantage of that. So, well, and of course, in the genetics of litter size and its components, we have been also examining things that we can, because as I say, it's cheap, the number of fetuses, the size of the placenta, the irrigation of the uterus, etc. So there is a lot of papers published about all of that type of things. What we are changing now, this is another, I put on a slide related to rabbits or whatever, to say that we are changing the topic. And we are now talking about genetics of resilience and longevity. I talked about longevity before, What we did is to compare a normal line with a standard longevity, with our Rambo line, our very, very strong line, with much higher longevity because of the founder effect that we have on this line. And the genetic for resilience was made with a divergent selection experiment for residual variants of litter size. What we are doing and we are analyzing now is in the line that has only 1, 2, 3 parities, until 5 parities, we take individuals that arrive to the first parity and died in both lines, and we take, samples of the feces of them. And we do the same with the individuals that arrive to the parity number 20. And we have that in the parity. So we have individuals that have first parity and died. So we have this information in the sensible line, in the sensitive line, and in the normal line. And we have the same with individuals that arrive very, very, very long, 20 parities. And we have information of their first parity and their last parity. And we are now trying to compare all of that, to find whether, and we have found some things, I'm not a microbiologist, and I'm usually worried about the results of we have found these metabolites and we have found this, but we have done all the omics on that. We're now analyzing the main results. Well, about genetics of resilience is what I say, that there are animals that are very constant and animals that they are not. So the idea is that if you have a more uniform litter size, you will have a better welfare and a better resilience. Which is great, because I didn't know that I was selecting for welfare either. I was selecting for, a problem that I was related to reducing environmental variability and improving heritability and things like that. But no, I know that I am working with genetics of welfare and resilience, which are successful words, I'm quite happy, we are working now in this. So the problem normally is attached like that. You say, I have a trait, for example, litter size, in which it has an additive value, and the variance of litter size has a function that can be depending on environmental genetic causes, and there are several models, and the genes that are controlling the residual variant, it's true. It's quite obvious. If you have litter size, you have an environmental variance of litter size, which means that there are animals that are more sensitive to illnesses. And illness, the appearance of an illness is an environmental factor. And some animals are more sensitive to that. And that has genetic reasons. So genetics is controlling the environmental variance. The same with stress and things like that, So there are animals more sensitive to stress. And this is controlled by genes as well. And these genes can be related to litter size, or to the genes of litter size. And they can have correlation. So the model is complex. They only can be solved properly using Bayesian techniques. And the problem is that it's very, very sensitive. For example, if you take the residual variance as is not exactly normal, but only approximately normal. Can you somebody, find me a bottle of water? Because I'm talking and talking and talking, and I'm a bit dry. I don't know how long away are the bottles of water, but no, the thing is that if you transform the variable and you put by a box-Cox transformation or whatever, and you say, well, now the residuals are pretty normal. Suddenly, the genetic correlation between the genes, considering the variance of litter size and the litter size, changed from minus 0.73 to plus 0.28. So the model is very sensitive to the assumptions you have. So you decided, well, why do not select? We have a meeting in Valencia with Dan Janola and Sorensen and people from INRA, and we were thinking, well, why do not select, just, by. Thank you very much. Why do you not select, directly, for residual variance? What do you mean with directly for residual variance? Imagine you have a female with several parities. And imagine that we can admit that the genes for all parities are the same, and only the environment changes and the fixed effects. Well, you can remove the fixed effects, by pre-correcting the data or whatever. We later observed that the results pre-correcting or not pre-correcting the data were more or less the same. But if the additive and permanent effects are the same, only the residuals change. So the observed phenotypic variance within, though, is the residual variance for lesion size. You're estimating that directly, with no model behind, And we started selecting 2 lines, for incrementing and reducing, litter size variance. I put 10 generations here. We're in the generation 14 now. Well, the idea is the same. We have genetic trends, but we have, at the end of the day, we have divergent lines of phenotypic differences. So if both agree, your model is corroborated, at least in the sense of Popper. You can corroborate the model because you say you obtain the same results by using genetic trends than when comparing. The problem with comparing is that you have much less animals in the generation 10 than considering the 10 generations together. So why it is important, to consider all lines. This is the phenotypic differences between lines, and it looks like we had success with that. And the residual variance, the difference between lines, were being bigger and bigger. And the genetic trends are more or less like that, So it looks like we have had success. The problem is that whether you have success only in one line or not. We are waiting for thawing the embryos to see whether the response is symmetric or not. But by the moment, apparently, the response is quite symmetric, approximately, symmetric, and we have had success in both lines. And litter size, of course, that was the great surprise. The homogeneous line had higher litter sizes. Why? Well, it's obvious. If you have a line that has 14, 15, 14, it has a small variance. If the line has 15, 8, 14, it has a larger variance. So when all the parities at the end were high, the line has a small variance. Of course, it happens as well with 5, 5, 6. But you have much more animals to select, and you can apply a bigger pressure of selection when you have 14, 15, 14. So finally, we are producing a line that can be useful for the industry, not only because the line is more homogeneous, but has a better litter size as well. And we comproved that it was not ovulation rate, but it was the number of implanted embryos which make the big differences between lines. So after doing that, we checked, the survival at birth and the cooling. And in both cases, the homogeneous line had better figures, for cooling and survival at birth. They were less cooled and have a higher survival at birth when you compare both. And if you take, for example, the zero-active protein, which is an indicator of inflammation, and you vaccine, and you check that before and after vaccinating, and you do the same with cortisol, that you put on a stressor like ACTH, and after the stressor, you measure the cortisol before and after the stressor, you see that the line selected for more homogeneous litter size has a better results. So it's more resilient to inflammation, and the same with lymphocytes, with inflammation and to stress as well. So it's interesting. So did we have success? Well, of course, if you don't have success, you change the definition of success, and that's arranged. But the response to selection is about 4.5 per generation, which is not bad, little size is not deteriorated, and the high lines is more sensitive, you know, to disease and to stress, and more cooled. And so the homogeneous line, which is the low line, could be perfectly a commercial line. So we're improving resilience, and if we're improving resilience, our argument as the animal is happier. Is an animal that is less ill and has less stress is a happier animal. So we are selecting also for welfare, which nowadays is important. Nobody takes care now about feed conversion rates, and so everybody's thinking about the welfare and about the conditions of the animals. Let me make a small interlude about what we are doing, about some theoretical developments that we were obliged to do. You know, when I started Bayesian analysis, I started, working with Robin Thompson, who I was working with him for 5 years, and it was hard to understand Robin, not only because of Robin, but because of the, the matters that we were working with, But when I met Dan Gianola, I thought it was like opening the window and the lights of the sun arriving and saying, oh, great, I see the things, at least I understand what I am doing, So we have in Bayesian theory, we have posterior densities to describe our uncertainty. The areas of these functions are the probabilities. So people did normally, or people working Bayesianists did the shortest interval with a 95% of probability, because the interval should be short. It's better to say that I think it's between 2 and 4 than to say that it's between 1 and 5, so it's better. And, but the problem with that, I was unsatisfied because we can do much more interesting things, For example, forget hypothesis test. Hypothesis test is a source of confusion. P-values, the only thing that they are confusing people, and are normally useless. And if you have a big sample, you have a small p-value, you have a small sample. Do you want stars? Take a bigger sample. Do you want NS? Throw a part of the sample. Intellectually, it's not very satisfying. So, but now we can say, well, why do not consider which is the probability of the difference between two lines or two whatever being bigger than 0? This is not a hypothesis test, but it's the actual probability of something, of some difference bigger than 0. And perhaps 92 is enough for you. There is no significance. There is no repetitions of things that I'm not going to repeat never. This is just the actual probability of the thing that we are observing. And I said, well, but the problem is that you can, how many times, in congresses and the speaker says, oh, we found significant differences here. And I asked, listen, but they were big or small, considering the type of trade you have. And sometimes the answer is, well, I do not know. Well, in that case, why are you dedicated to science? You can work, making massages, or football player or whatever. But if you are working in science, it's supposed that you should know about whether the things that you have in hands are big or small. So the thing is not whether you have a difference or not. It's whether this difference is relevant enough to take a decision. So why do not put in papers, instead of the significance of probability of being 0, the probability of relevance? Which is a relevant value? Well, a relevant value, when you design an experiment, you design how many individuals do you need for significance? Well, this is the relevant value. For this value, I will need that number of individuals with significance. It's the value, the minimal value to take a decision. So you can take probability of relevance, or you can say, well, for example, a correlation is never going to be exactly 0. 0.0000. But if it's lower than 0.1 or minus 0.1, probably this is enough for you for saying that it's 0 for practical reasons. So we say that there is no difference, but for practical reasons, there is no difference. The difference is irrelevant. And there is no difference to take a decision. With 0 for me is to be between relevant values. That is much more clear than 0, 0, 0, 0, etc. Things are always different from 0. So I didn't know whether that was right, but we had a very powerful team in my place, about Bayesian people working with that, and they say that it was correct. We could work with that, and we did. The guaranteed value, the minimum value for a guaranteed determinate. So all of that is in a book that I recommend you. I do not recommend you to buy the book. There are many pirate, versions of it. So then we worked with omics, and we had a similar problem. With omics, that the problem is that if you augment or reduce, you do not have compositional currents. The percentages change. If you add numbers that are not percentages, the rest of the variables are the same. But if you add numbers, add a new variable, for example, the percentage changes. So people do not know what to do, and it was suggested to work with ratios. But with ratios, everything are the same. Which is the main problem with working with ratios? The main problem is that I do not want to work with ratios. It's very inconvenient. I do not like it. So we proposed at first to use low ratios and to use compositional incurrent. Well, ELR is the ratio between a variable and a reference variable. And we wanted to use the reference variable with less variance. Because if it has a low variance, we can, just think in the numerator only. And we discovered later, it's complicated, that the variables, we did an approximation of our system to the whole system with log ratios by Procrustes analysis and things like that. And we discovered that the result were more or less the same. So our system was almost isometric, and we could use that. Now, the thing about compositional data, if you work with that, is that we have a lot of zeros. And the log of 0 doesn't exist, and people impute, things to zeros, or use Mickey Mouse priors. That is a thing that I will tell you later what I mean with that, So nobody knows what to do with the zeros in omics analysis. And well, we have recently obtained the best solution, because all of the solutions that has been proposed are unsatisfactory. The best, what we did is to take a database of microbiome and produce several sub-compositional data, keeping, the main microbes as main microbes and things like that, and we did all types of analysis with them. We compared all the things that you can imagine or have in mind: Procrustes correlation, PLS, random forest, and bloop, and whatever. And we obtained the solution for this type of problems. The first is that you can use our system of taking, ELR, using the variable with the least possible variability in the denominator. But actually, it's not necessary. The best thing is to do nothing. The best thing is to just analyze the data as they are, and you obtain the same. I remember when we started analyzing microbiomical data, we went to the capital of microbiomical data, which is in the north of Spain, and there is a big group there working on that, of mathematicians, and we told them, listen, we know that in compositional data that assume to 1, if one variable goes up, the other goes down, and things like that, but when I have 6,000 variables, probably the effect is diluted. They told us, we cannot prove that, and if you do not have the proof, you have to... Listen, we have proved all type of analysis with data of cows, rabbits, humans, with all omics, and with all possible analysis done, and we always permanently have obtained the same results. So liberate from the chains of the log ratios, forget imputations, and be happy, analyze data as they are, and this is our great last discovery that is going to be published soon. Well, there are a couple of, theoretical incursions. Let's now change the topic, and and go to another topic now, which is the genetics for field efficiency. I selected a line for growth, and the thing selecting for growth is easy. It's not only selecting for components of liter size. But here the challenge was that the growth curves are double exponential, and a part that are e to the exponent to 0 to the exponent is that these parameters have fixed and random effects. And even worse, they are correlated. And even worse, this is what we obtain in practice. So at the beginning, the animals are all small, and later they grow, and later they are all big. So the residuals augment with the age until they are stabilized. So the residuals have their own growth curve with their own parameters. So we have to estimate the parameters of the curve and the correlations and the residuals. And we did that, of course, by using Bayesian techniques, the only proper way for doing that. Well, what we observed that was funny, my friend Killier Taylor told me, well, if you represent that using the metabolic scale, which is using the scale considering metabolic weight, you will see that both curves are the same. Because selection, the only thing that you did is to augment the whole curve because all the weights are correlated. So it's not the most efficient way of working. Well, about our new line for efficiency, we have created that, by blending commercial lines, pure lines, there are no pure lines, and the selection criteria was discussed. Residual food intake, consumption, and I must admit I am the only man in the world that doesn't understand what residual food intake is. And as I don't understand that, I say, well, first, it's not a residual, it's an estimate of a residual. And the residuals are uncorrelated, the estimates are not, but apart from that, many things. What do you study when you are in the agricultural school? Where the money comes from? From the rabbits I sell, okay, weight of the rabbits. Where the expenses come from? Most of them from how much food the rabbit has eaten. So just make an index, selection index for food and weight, and that's it. And I do not need, this complex thing saying that we are conserving the. If some of you understand residual food intake later, please try to explain that to me because I don't. Well, and let's change as well to another topic. Genetics of carcass and meat quality. Well, these are common words. We selected for intramuscular fat in rabbits. Intramuscular fat in rabbits is not very high, but it's enough. It's 1.07, 1.10, or something like that. So we selected, and we can compare the selection at the end, as usual. And we know that the differences are due only to genetical reasons. So we estimated correlated to all omics. So we could see, how the omics are affected by selection in the best possible way. We have two different lines that only differ by genes because the environment was the same, and we are comparing them. And there is a lot of romantic literature talking about all of that type of things. And I will end with my personal career. This is the funny clothes we have for doctoring in Spain. This is me when I was, how old was I? 25, 26 or something like that, 26, I think. I was an agricultural engineer, and I became quite quickly associate professor. Well, associate professor is, I mean senior lecturer. I don't know exactly how you use the words. Senior lecturer quite soon, and full professor quite soon either. And I had a sabbatical at what is now called Roslin, another year in Paris. I never do sabbatical years in places that do not have a good opera theater. So I went as well to Rome, to FAO, for 6 months. And I've been the head of the department of the institute of our masters that we have. I was the president of the World Rabbit Science Association. There are associations for everything in the world, and one is for that, An editor-in-chief of World Rabbit Science. I was Chapman Lecturer in Wisconsin, and I have an Spanish National Award, and I also Georgophilic person, because I belong to the Academy of Georgophilic. When I told to my wife that I was a Georgophilicus, she thought that it was a sexual transmission illness or something like that, to be a Georgophilic, but no. It's the oldest academy of agriculture in the world. It's a prestigious academy in Italy that has some external correspondence. And I have given a Bayesian course at the university in English, in Wisconsin, in Edinburgh, in Italian, in Milan, and Udine, and Padua, in Portuguese, and São Paulo, and Lavras, in Spanish, in Montevideo, and in Spain, of course. And I didn't do that in German, because with 3 years of Italian, I can read Petrarca, but with 3 years of German, I hardly can ask for a sandwich and a beer. I published a couple of books, about things related to our field, and a book about animal ethics as well, because all of these books of animal ethics are written by people working in moral philosophy. So they do not know what a biological animal is. They think that a cow is like us, but less, things like that, But so it was important, to have a biological perspective on that. And I did that because I was the president of a committee, for improving the welfare of rabbits and so on. I have created a net for communicating people speaking Spanish and working in animal genetics. And the reason is that for us, to speak Italian, Portuguese, and French, it's easy. It's just corrupted Latin, But if you want to speak English, it's more difficult for us, So people are feeling insecure. They do not take part in forums, and so because of insecurity. And even a good friend of mine, Miguel Ángel Toro, once proposed a course on animal breeding. But instead of a course, he put a Spanish word, C-U-R-S-E, a course on animal breeding. So the answer was sooner or later that was going to happen in a course on animal breeding. It was a bit of, we said, well, let's create a net in which you could speak Spanish and we can understand better each other. Now the net has about 500 scientists along the world, from Alaska to Australia. In Australia, there are people also belonging to this net, and we are putting congresses, software, things like that, and several things. And now I finished, a couple of things about me, because the organizers told me as well, talk about, your life and what you have done. And well, first, I have a boy who is mentally handicapped. He's 28 now, has a 67% of mentally handicapped. And I am the president of the Valencian Association of Children like him. He's mentally handicapped, but he can play piano. I mean, he plays, Bach, and the thing is, why do you think that? Well, the thing is that we are working to be. It's like if you have a friend with a cancer, you do not introduce him as your cancerous friend, but as a friend who has a problem. My child is not handicapped. My child has a handicap, which is different. So the interesting thing is that we had to develop all the things that in developed countries they have, We developed finally units of languages for them, and we developed, we are starting with the flats supervised for them and things like this, And apart from that, I play piano and play harpsichord. I like that, both, and classical guitar, and we make concerts in agricultural school once per year, in which teachers and students that we can play classical music, we play classical music concerts in there. I also have written some books, one book of short stories, fiction stories, a book of formal poetry that cannot be translated. There are sonnets and octaves and things like that, Formal poetry is impossible to translate, A book on essays, and a book which is my Roman diary about my life in Rome, very much in the style of the Italienische Reisen of Goethe. But the thing that has had more, my hobby that has more impact in our field is ballroom dancing, because this is the World Congress on World Congress of Rabbit Science in 2000, in which, as a part of a spectacle, there were some dancing and so on. Finally, my wife and me went there to dance a tango. We have been going to classes in the last 35 years, and we still go to classes every week to dance on Saturdays, It's an activity that I recommend you, not only because of the physical benefit, but only because of the mental benefit. With the time, you have told to your couple, everything that you can tell her or him, at least, and to dance is to be embraced to a person that you love for a couple of hours without the need of keeping on the conversation. So that's a great activity that I recommend you, to do that. And well, that's it. I finish with my talk. I'm sorry about the interruption that I had.
That's right. So this seminar is recorded, and so there you can follow this QR code to look at the collection of other seminars. And I think that's about it, correct? Awesome. So one more round of applause for listening.